Hey, Hammy here, back with part one, or the first part of section 3.2, where we begin to get into all the little pieces and parts of the cell. The cell, eukaryotic cells with all of these different organelles, we often say resembles a bag full of jello. Okay, so it's kind of this liquidy fluid, and some of you have done those cells in a bag type projects in elementary school, okay, where you have kind of a container that is holding all these little pieces and parts, and they're all kind of can move around and that kind of stuff inside the cell. So we want to start beginning to look at what are some of these pieces and parts of the cell. First, we have to talk about the plasma membrane, okay, or sometimes we call it the cell membrane. Okay, that is the barrier or the thing that goes all the way around the cell, okay? So it's kind of the outside. It would be the Ziploc baggie that you would have done your cell in a bag project, okay? So you have over here, if you look at the diagram over here, okay, so here's the cell, and you see this yellow line here. It kind of, you have to think of it in 3 dimensions. It goes all the way around the cell, but it's not just like a plastic liner. It is actually very dynamic with a bunch of different parts that we're going to look at here in a second. And the important thing about that is it helps protect, support the cell, give it its shape, but it controls what enters and what can leave the cell. Because it lets some things in and out, but not others, we often say it is semi-permeable, or sometimes you'll see it as selectively permeable. Okay, you need to be familiar with those two terms, semi-permeable and selectively permeable, only letting certain things in and or out of the cell. When we really zoom in on the cell membrane, really, really close, again, I said it's not just this plastic boundary. It's a very dynamic phospholipid bilayer. Now, you should remember from last chapter, the chemistry chapter, what a phospholipid is. Okay, it's got a phosphate head. Okay, right here you can see the P or the phosphate group, okay, with 2 fatty acid tails, a glycerol backbone right here, and a phosphate head. Okay, that makes the phosphate head has these little charges in here. So we say it is hydrophilic. Hydrophilic means water loving. Okay, so the heads you can see down here on the bilayer sheet, the heads face towards the outside and towards the inside. Okay, this would be outside the cell. This would be inside the cell. What's going to be out there? Mostly water. What's going to be the cytoplasm? Mostly water. So the heads are water loving. They'll face outward towards the water, the interstitial fluid, and the cytoplasm on the inside. The tails, because they're just carbon to carbon to carbon to carbon bond, that is hydrophobic. Okay, they're not polar. They do not like water. So all the tails are going to be in this middle region in here because they do not like the water in here and out here. Okay. Also, if you look at a close-up, you have this. If you flip to the next page on your notes, you have this. Would be page 4 of your notes. I want to highlight certain things here that you're going to see this diagram again, and I want to point out some things you'll be responsible for. Okay. First of all, understand this is a phospholipid bilayer. So there's 2 layers of phospholipids. Okay, you should be able to recognize one phospholipid molecule. You should know that the round part is the hydrophilic head. The tails are hydrophobic tails. You should know that these little yellow things stuck in here, okay? These little yellow things stuck in here, right here. I don't see them. Oh, yeah, over here is the label. Okay, they are cholesterols. Okay, so these little yellow chain kind of, remember, they're like steroids, which would be 4 fused rings. The cholesterols give some kind of stickiness to the phospholipid bilayer, kind of help hold things together. You need to understand that there are these blue blobs here, or proteins, that are stuck inside of here that have different enzymatic functions. Also, some are open in the middle, so protein channel, how certain things could get in and out of the cell. You'll notice that there are these little carbohydrate chains. So these little chains here off of either proteins. If you have a carbohydrate chain off of a protein, it's called a glycoprotein. Sometimes they can come off of a lipid, so you can have a glycolipid. Okay, and those are used for cell recognition, and we all have different chains and that kind of stuff on the outside. Sometimes they're receptors. You'll learn about receptor-mediated endocytosis. So these often play important functions in the cell, recognizing materials from the outside. Okay, so you need to know these different pieces and parts that I have underlined and circled here on this diagram. The plasma membrane can have extensions called cilia and flagella. Okay, here's a little algae, okay, where you see the flagella hanging off of here that will help it swim through the water. Over here is an example of cilia. They're more like little hair-like extensions. We have cilia in our throat so that the dust and stuff we breathe in our trachea, it sweeps it back up, and then you can swallow it so that it doesn't get in your lungs. This is why smoking can be bad, because it deadens those cilia. Excuse me, and the dust and stuff falls down in our lungs, okay, which can increase the risk of things like black lung and cancer and that kind of stuff. So there are extensions of the plasma membrane that help a cell to move or stick to surfaces or move stuff outside of the cell, like in our throat. The second part we want to talk about is everything inside of the cell membrane, which we call the cytoplasm, or sometimes you'll see the word cytosol, okay? Oops, which is also one of your blanks there. This is everything inside the plasma membrane. Most of it is water. It kind of has a jelly-like, so when you did your cell in a bag, you may have put water in, or some teachers have them put like a jello or gelatin in. That makes it a little more realistic. Okay, so it's kind of a watery gel-like substance. And it will contain all these other structures that you see over here. Okay, so all our organelles, nucleus, all that stuff will kind of be suspended in that gel. But again, remember, it's mostly water. The purpose of the cytoplasm is to, again, it's a place for the cell organelles to kind of be stuck. It pushes, in some cells, it pushes against the plasma membrane, helping to keep its shape. Okay, especially in plant cells, turgor pressure, that's important. And we'll talk about that later this chapter. And also, the cytoplasm is a site for our metabolism. It's a place for a lot of those chemical reactions that take place, whether it's in an organelle or it's just happening in the cytoplasm. It's a site for all those chemical reactions. And finally, we have our cytoskeleton. Just like we have a skeleton that kind of gives a framework of our bodies, cells have a skeleton that kind of gives them their framework. There are thin thread-like sort of tubes called filaments, and then there's thicker tubules. Okay, these are both protein-type sticks and tubes that kind of pull and stretch and kind of run and make this meshwork network throughout the cell that we call the cell cytoskeleton. It helps give the cell its shape. It holds the organelles in place, because you're like, well, we talked about the cytoplasm just being jelly-like. How can the organelle just stay right where it is? Well, it's held in place by the cytoskeleton. It can help the cell move. Things like amoeba will stream, okay, where you have, maybe here's an amoeba cell, nucleus, and it sees a little bacterium here that it wants to eat. It can actually stream the cytoplasm out both ways here to form these, what we call pseudopodia, or false feet, and will kind of ooze and surround that cell, and eventually it'll pinch off here and it can take that little bacterium in to digest it for food. Okay, so it helps cells to move. The cytoskeleton moves things as proteins and stuff are made. It moves it from one organelle to the other as it's processed through the cell. So it will move stuff within the cell. And it also helps with cell division. When we talk about cell division later this year, we'll talk about these spindle fibers, and you can see that happening over here in the picture. As the two cells kind of push apart from each other, the spindle fibers kind of help stretch the cell apart, and they also help grab the chromosomes and pull the chromosomes, sister chromatids, in opposite directions and help divide up the genetic material to the two new cells. So the cytoskeleton plays an important role for that. Okay. Now, there are a whole lot of other cell parts we're going to talk about, but we'll continue that in the next video.